or chicken, the maximum percentage of protein is 20 to 22 percentage. Whereas if you are using this chlorella, it's a microalgae, you may get more than 55 percentage of protein. And if you are utilizing the spirulina, it is a blue green algae. In Tamil, it is called surulpasi. It has more than 71 percentage of the protein. It is the only bioresource having the highest percentage of protein. So this is the very excellent candidate to fight the malnutrition in the undernourished countries. Then coming to the aqua feed, as a primary producer, definitely it acts as aqua feed. So in our department, during my teacher, Dr. G. Subramaniam's period, he developed one aqua feed from the cyanobacterium called Formedium multirianum, and he transferred the technology to a company at the cost of nearly 50 lakhs. So this is possible. Then these potentials of other microalgae, because it has some autofluorescent pigments, we can utilize these pigments, which is alternate to isotopic labeling. So we need not use any isotopic labelings in the immunodiagnostic kits or medical markers or any chromophores for identification purpose. We may use this pigments like phycocyanin, phycoerythrin, carotenoids, and chlorophyll. These are all autofluorescent pigments we can utilize in the immunodiagnostic kits. Coming to the restriction enzymes, in molecular biology, this is the very costliest products available in the market. But some of the cyanobacterium like AVA1, Anabina variabilis, one restriction enzymes is commercially available in a very cheap cost, because the cost of production is very, very less when compared to the same product developed using the bacteria and fungi. We have to have the defined medium, sterilized condition, and so on. Whereas here, growing the algae, we need water and sunlight. If you want a higher biomass, you can add the urea and the superphosphate in very minimal quantities. So we can get the huge biomass out of these chemicals. And another important component is the siderophores. Siderophores are really the ion binding chelators. So it is medicinally very, very important, because if any person having the higher iron content, it is lethal to them. So if any person having the higher iron content, if go to the doctor inject the medicine called diffroxamine. So diffroxamine is nothing but the siderophore. So if the siderophore injected in the blood, so it scavenges the iron content in the blood, it forms the iron siderophore complex. Once the complex is formed, so there will not be iron. So our immune system try to expel this siderophore iron complex from the body stream, blood stream, so that we can minimize the blood level, blood iron content. And another most important point is the bioenergy, that is biofuel. So we can generate hydrogen, diesel, biobutanol, and so on. So almost all the energy requirement can be fulfilled by the microalgae and cyanobacteria. And now all the countries, the scientists are recommending and accepting that the microalgae is the only best resource for the biodiesel or bioenergy production. It is the sustained resource for getting this different kind of energy. And coming to the phycobiliprotein, as I said, the phycocyanin and the phycoerythrin, it is autofluorescent. It has a very excellent coloration also, red color, blue color, and so on. So we can utilize this. So whatever the food colorant we are consuming through food or sweets, in long term it is carcinogenic. Whereas the phycobiliprotein, the phycocyanin, phycoerythrin are only proteins. It is plant proteins, easily digestible. It has the high nutraceutical value. So we can use this. Again, we developed the phycocyanin technology, that is blue colorant technology, and transfer the technology to company at the cost of nearly 50 lakhs again. And nanoparticles, now almost all the researchers from even the engineering discipline and science discipline are about to develop the nanoparticles, because the specialty of the algal nanoparticle is so if you want to have a triangular type of gold nanoparticle, you use one algae. If you want hexagonal type of nanoparticle, you can use some other algae. Likewise, whatever size, whatever shape you want, you select the different type of algae and we can get the desired products. So this is also possible. We published several papers on nanoparticle synthesis. Then coming to the toxins, yes, algae or cyanobacteria are producing toxin. Not all the algae or cyanobacteria are very beneficial. Some harmful microalgae, harmful cyanobacteria is also there. Now, toxins are also equally important in the view of developing the bio weapons, as well as the toxins in some proportion are useful in treating the cancer. So some proportion of the toxin, it kill only the cancer cells, not the normal cells. So likewise, the research is going on. Then coming to the antibiotics, no doubt. So almost all the cyanobacteria and microalgae is having the antibiotic property against any clinical isolates. So the intensity or vibrance of the antibiotic activities differ from species to species, but it has the antibiotic activity. Then coming to the fatty acids. Fatty acids important for the biodiesel production. In addition, some of the fatty acids like gamma linolenic acid, that is GLA, it is medicinally highly important, and it has the crucial role in contraction and repulsion of the heart muscles. So in some of the hypersaline cyanobacteria, we reported the higher quantity of the GLA in the cyanobacteria. The coming to the vitamins, no doubt, we have the amazing quantity of the vitamin content in the cyanobacteria and microalgae. If you go to the Google search, you just type the vitamin content of any other plant or animal resources and compare with the vitamin content of spirulina, that is blue green algae. Really, several lakhs units of vitamins are present in these organisms. And another one is most important that is need of the hour is the bioremediation process. Our country is always in a developing country stage because of the explosive population, industrialization, and the emanation of the effluent through the environment. So it is very difficult to stop the effluents, difficult to stop the industries. But we can bioremediate all kind of effluents using the microalgae and cyanobacteria because the hypersaline cyanobacteria we isolated in our laboratory, they can be used to treat all kind of effluents. We are getting the 100% success. So in subsequent slides, I'm going to show some of the data on bioremediation. And finally, anti-fertility compound. Still, our country is a developing country because of the explosive population. As I said, to check the population, usually the birth control, the women are victimized. Now, one of my colleague, Dr. Tirunala Sundari, she collected some cyanobacteria called Oscillatoria villi species. She treated the extract of Oscillatoria villi to the mice and some lower animals, and she found that there is a 80% of the sperm reduction. So remaining 20%, the sperms are abnormal, headless, tailless, multiple heads with a single tail, then multiple tail with single head. Likewise, the total malformation, we can proclaim that there is 100% sperm reduction. So coming to our marine cyanobacterial survey initiated during the year 1987 as a PhD scholar worked under Professor G. Subramanian's project on marine biotechnology, the cyanobacterial biomass production and application. We surveyed the cyanobacterial wealth of our country starting from Nagur to Kanyakumari, and we identified so many marine cyanobacteria for the first time in our country. And out of our PhD work, we established a culture collection, germ plasm collection of cyanobacteria. Later on, this germ plasm collection is converted to a national facility for marine cyanobacteria. It is only sponsored by DBT, Government of India. So when we are going for the biodiversity, biodiversity is the base for any biotechnology. The basic and fundamental requirement for initiating the biotechnology of any kind of organism, we must know the available wealth of the biodiversity and try to isolate, purify, and maintain the cultural collections. From the cultural collections, we can exploit or utilize their potentials for our benefit. So if you are going for marine cyanobacterial biodiversity, so our environment are greatly influenced the biodiversity of microalgae and cyanobacteria. For example, this is the sandy nature of the seashore, this is the muddy nature of the seashore, and this is the rocky nature of the seashore. And the seashore of Kurusra Island, it is completely made up of coral reef, occupied by the coral reefs. So among this Mantapam Rameswaram area, that is Pamban area or Gulf of Mannar area, it is we are considering as algal paradise. So because almost all kind of flora and fauna available in this type of environment. And this is the another very potential environment called salt pans. That is hypersaline environment where you can see the salt crystals. Below that, you can find the algal bloom. So, in drinking water, there is no salinity. We call it as 0 ppt, that is parts per thousand. Whereas in seawater, it has 35 ppt of salinity. Whereas in this kind of salt pans, more than 200 ppt of salinity is there. So our organisms are very luxuriantly growing in a very high stress condition. These organisms are the efficient candidate to bioremediate all kind of effluents. So if we lose any kind of effluent, we can get it. Someone making the mark like this to avoid this kind of disturbance. Okay. Otherwise, I close my lecture. There should not be any disturbance from this audience. Okay. So we then later on, after establishing a national facility, we started surveying from marine cyanobacteria from Goa to Kanyakumari and Kanyakumari to Bhimanipatnam of Andhra Pradesh, and covered Andaman, Nicobar Islands, Lakshadweep Islands, and several Gulf of Mannar group islands we selected. And we surveyed, we got very excellent data, not only the data, the culture collection, that is in marine germplasm, we established in Bharadasan University. Now almost all the students who want the cultures of marine cyanobacteria can send the request through the email. They can get the cultures in a very minimal cost. And this is our overall biodiversity of marine cyanobacteria. 240 species we identified from the South Indian as well as Andaman, Nicobar, Lakshadweep environments. I'm not going to detail about it. This is the marine cyanobacterial germplasm culture collection in National Facility for Marine Cyanobacteria. Anyone can access this culture collection and get these cultures for their research work or PhD work or any project work. It is available. And this is another paper we published by invitation, this cyanobacterial biodiversity and potential applications in biotechnology. So we published with very excellent photographs, and this paper cited more than 500 citations we received for this current science paper. And so far we studied only on marine cyanobacteria. After 2006, so I shifted from Marine Cyanobacterial Center to Department of Microbiology as head of the department. I started working on the freshwater microalgae and cyanobacteria and developed the germplasm collection. Later, recently, we established the second national facility, that is the National Repository for Microalgae and Cyanobacteria, exclusively for freshwater, sponsored by DVT Government of India. We constructed a separate building also. You just imagine the basic and fundamental work, that is the enumeration, identification, and establishment of culture, leads two national facilities of our country in Bharadasan University. And wherever we go, we for biodiversity survey, in the field itself, we sit and identify and take the micro photograph of all the microalgae, algal diversity in the field. So this is very important because when you are collecting the sample from any seashore and bringing to the university, it will take nearly 5 hours. So during the 5 hour, the zooplankton or the other animals, micro animals, protozoans that eat or consume almost all the biodiversity of microalgae and cyanobacteria. So to avoid this, to record the 100% diversity, we sit in the field itself and take the micro photograph of all the biodiversity so that we claim that we did the diversity work very perfectly. And these are all these few cyanobacterial, that is microalgal culture collections we are now established from our survey work from Class Stadium. Very attractive, beautiful structures we have in our germplasm collection. and you can see this very beautiful different structured diatoms also. So these organisms are very important for biodiesel production. And you can see the beautiful view of Senidismus. So this is the confocal microscopic view. The confocal microscope, cost of the confocal microscope is nearly 1.2 crores. So here we can, this is the cross-sectional view without cutting the cells. So we instruct the organ, through the software. If you want 100 cross sections or 200 cross sections or 50 cross sections, you just type instead the computer system attached in the confocal microscopy, you are getting the cross-sectional views. And also we can manipulate the color also using different filters. So these are all the different filtered microalgal photograph assembled in a single view. Senidesmus, this is green algae. And this is again the confocal view of Aphanocapsa species, that is the cyanobacteria. And this is another Crococcus species, two-celled and four-celled structure in different color manipulations. And this is again Gliocapsa species. This is again cyanobacteria with different colorations. So these are all very attractive photographs. Nowhere it is available. It is available only in our national facility website. In addition to the freshwater, we have a collaboration with the Antarctic Center at Goa. We published a paper on Antarctic biodiversity and distribution of cyanobacteria in the East Antarctica. So this is, we are getting very good data on the Antarctic cyanobacterial flora. And as a result of our freshwater survey and identification, we again developed the freshwater microalgae in cyanobacteria. And later, DBT invited us to submit a proposal for establishing the freshwater national repository, and we are successful, and we developed the national repository in Bharatdasan University. And coming to the application, so far I mentioned some basic biodiversity aspects. Yes, biodiversity, since we are one of the 14 mega biodiversity countries of the world, we have richest population of microalgae, cyanobacteria, fungi, flora, fauna, insects, and so on. So only thing is we have to identify and develop the germplasm, and then we explore and utilize for our benefits. So among the development of the biotechnological development of the microalgae and cyanobacteria, first foremost organism is the spirulina. Spirulina biotechnology, because it has a very excellent protein content. If you see this slide, this is published in this obtained from the literature standard publications. You see in the rice there are 7% of the protein. You fish 20%, beef or mutton or chicken 20% maximum, eggs 25%. So egg is better than the mutton or chicken. Then soya bean, it's a plant source, maximum 35% of the protein. In chlorella, it is a green algae, maximum 56% of the protein, whereas in spirulina, maximum of 71% of the protein, we can get it. So this is the very unique features of the microalgae and cyanobacteria. And even in the African countries, people directly harvest from the open natural ponds, and then without cooking, they are consuming the spirulina, natural spirulina, because there is no toxicity, there is no side effects. It is 100% nutraceutical value and also some medicinal value also with this spirulina. Even self-help group, they are growing this spirulina in the tubs, plastic tubs, and they are now exporting this spirulina to the other countries. And this is the important slide I like to share with you is a few years back I gave one spirulina entrepreneurship lecture in one Pannaya Ramajayam College, where this student, Mr. Thanga Prabhakaran, he was the first BTech year student, and he attended my lecture, and he developed interest on spirulina cultivation. Then he came to my laboratory and requested me to get some knowledge, hands-on training on how to grow this spirulina, what are the problems we are facing, likewise at the laboratory level and the pilot scale level. And I gave some brief training of nearly 15 days. To my surprise, in the first year, he asked the questions and I got trained in my laboratory. And in the fourth year, during his completion of BTech degree, he established a company called Elixir and released the spirulina product also. Now he is a millionaire. He received orders from UK and other countries. Recently, he received 6 crore worth order for the supply of this spirulina. Now you just understand the importance of these tiny organisms. And this is his raceway pond he developed because of his engineering knowledge. He developed a type of raceway pond and developed the spirulina culture in a large scale. And he developed the capsules and other tablets also using the spirulina. And this is the capsule called Neutralite. It is available in almost all the medical shops in Tamil Nadu as well as in Kerala. So in addition to the other potentials file now, the organisms are very active scavenger or sequesters the carbon dioxide from the atmosphere. For example, if you grow the algae in 1 acre farm, 1 acre area, if you grow the algae, it sequesters 400 metric tons of carbon dioxide from the air. So you can easily reduce the global warming, and ozone hole is also reduced. Like this is the very potential organism. It is superior than the higher plants. So then my first PhD student, Dr. Nagasatya, she completed in 2005. Her work is in the bioremediation of hypersaline cyanobacteria in different industrial effluents. She isolated one particular species called Formidium tenue, KMD33 species. That is, she isolated from the Kattumavadi region of the Pudukkottai district. And she treated with a various effluent and found that it is very active decolorization of the effluent, reducing the BOD, COD and other elements, increasing the dissolved oxygen and increasing the nutrient content. Then whatever pH is there, whether it is acidic or alkaline, it converted into the neutral pH. So after treatment of these effluents using the cyanobacteria, we can utilize this water, treated water, to the fisheries as well as the irrigation purpose. So she published some good number of papers in international level journals, and this is how she developed the technology. Okay, this is the Hindustan Paper Mill Limited effluent collected from the Thiruvananthapuram. She treated with this algae. Now you can see this. There is 100% color reduction. So it decolorize the effluent, and also it reduces the BOD and COD and so on. So another PhD student, she worked on oil degradation using the cyanobacterium, the same cyanobacterium, Formidium tenue, KMD33 species. So this is the microscopic view. This is the microscopic view. Initially, the algae, the oil is in a very globular structure because of the high surface tension. So if you are, if my student is inoculated the cyanobacterium with the oil, the secondary metabolites released from the oil, cyanobacterium degrades gradually the crude oil. So you can see apparently the gradual degradation of the crude oil in the environment. And in the crude oil, there are two toxic substances are there. One is anthracene, another one is naphthalene. So if you are if you are inoculating the anthracene and naphthalene separately with the Formidium KMD33, we found that the anthracene is converted to anthracene dione. Naphthalene is converted to naphthalene dione due to the algal treatment, cyanobacterial treatment. So from toxic to non-toxic form. So we published this paper in biotechnology letters from US, and it has a very good impact factor and a very good citation also. I don't know who are giving this kind of red markings. So this is not really fair. When I am presenting some participant, they are making red marking in my slide. So you identify that person, and then you can give some instruction. So you have the
Okay, sir.
educated people.
Okay, sir. I will check it out, sir. Sure.
Yeah, this is irritating me. And the another type of work, we use the algae for biodegradation of the dyes, particularly rhodamine B dye. Initially, the rhodamine dye is a very complex structure. When we are treating with the microalgae, there is cholesterol, based on the secondary metabolite released, it degrades the complex structure and we can get some broken fragments. This is the GCMS data, and we refer to the library. All the broken fragments are non-toxic. So there is a bioconversion of the rhodamine dye using the cholesterol. It is a green algae. And this is the experiment. Initially, the rhodamine dye. This is the algae. If you treat this, you can see this degradation of the dye. So the candidate involved is Dr. E. Baldev. He published this paper in colloids and surface B interfaces, which is a 4.4 impact factor journal. He published, and this is the paper he published with a good impact factor journal. And in addition to this, we use the hypersaline cyanobacteria. We published a large number of papers using these organisms, hypersaline cyanobacteria, for the bioremediation of all kind of effluents. Like the journals like Journal of Environmental Sciences, Biotechnology Letters, Bulletin of Environmental Contamination and Toxicology, Annals of Microbiology, all good impact factor journals we published using the hypersaline cyanobacteria for treatment of the various effluents. And coming to the agriculture, so as I said, these organisms are very important for in biofertilizer, particularly the nitrogen fixation. In addition to nitrogen fixation, the cyanobacteria is liberating the growth promoting substances which enhance the agricultural productivity. To prove this, whether the cyanobacteria is liberating the hormones or not, to prove this, we use some tissue culture techniques. In the tissue culture technique, people using researchers using the MS basal medium with the addition of some growth promoting substance like gibberellic acid or indolacetic acid for shoot growth, shoot and root growth. And this is the first one. This is the MS basal medium. There is no hormone is added, and we inoculated the tomato seeds. We found the growth. And this is the MS basal medium with hormone, that is gibberellic acid. You can see this growth. So compare this growth without hormone with the hormone. And here in this third one, this is the MS basal medium. So we are not adding any hormone, but we add the 0.5 percentage of the extracellular product of the Oscillatoria species. So we grow the algae and we are not disturbing the algae. We are collecting the surrounding medium and extract the hormone, and we treat the hormone at 0.5 percentage. You see the vibrant growth. And this is again 1% of the extracellular product. You can see the difference in growth. When compared to the hormone treatment, the cyanobacterial extract is showing a very vibrant growth. This is the 25-day-old cultures. So we forget and we see this culture again in 40th day. The cotton plug in the top is thrown away from the flask, and all the plantlets are happily coming out from the flask. So such type of vigorous growth is there. So from this, we can say that we need not spend money for purchasing the gibberellic acid or indolacetic acid or any other hormones. We just use the extracts of cyanobacterial extracts, and we can utilize and we can get the very excellent growth. And in addition to this Oscillatoria species, we use a different cyanobacterium called Formedium, Chylothrix, Cytonyma, Nostoc. So we are getting the same kind of results. So from this, we proved that all the cyanobacteria are having potential of liberating the growth promoting substances. We tested with some green algae like chlorella and Celidesmus. We could not get this kind of result, and we say that the green algae is not liberating any growth promoting substances to the environment. And based on our research, we published one paper in Journal of Genetic Engineering and Biotechnology. These are all the two persons involved in this kind of research. So this is the collaborative work. As a microbiology, we collaborate with the botany department. We can get the excellent data as well as the very good publications out of this. As I said, the siderophores, importance of siderophores. So this is one organism called Oscillatoria salina species. It is very dominant in almost all the marine environment. So these organisms are liberating the siderophore only under iron minus medium. If it is iron plus medium, the organism will not produce the siderophores. So it requires the iron free medium, so that for scavenging the iron, it liberates the siderophore and to scavenge the iron for their growth and development. So this siderophore is again very important for the establishment of algal blooms. So algal blooms, initially if any pond, if you survey, there are more than 200 different types of species of microalgae, cyanobacteria, protozoa, and so on. So once the bloom is formed, only one or two species are dominant, and other species you cannot see it, totally vanished. So this is because of the hydrophores. So in hydrophore, in any pond, freshwater pond, the organism liberating the hydrophore and it scavenges the iron content in the environment and binds the iron, form the iron hydrophore complex. Once the iron hydrophore complex is formed, it is no more iron. The other organism could not get the iron nutrient. So the organism releasing the hydrophore can only take back the iron hydrophore complex, and the hydrophore joined in the cell surface. Only iron content is enter into the cell for the development. So this is one of the factors for establishment of algal blooms in the natural system. So this is another very interesting work we carried out under DBT project. That is anti-quorum sensing compounds. So anti-biofilm establishment using the cyanobacteria. So in any aquaculture environment, there is pathogenic Vibrio species is causing the biofilm. Once the thick biofilm is formed, it lethal to the fishes. Initially, in one or two or few cells of Vibrio species attached in the surface of the fishes, and it liberates some quorum sensing signal to the environment, and it attracts the quorum sensing signals, attracts the similar kind of pathogenic bacteria to the fish surface and forms the extensive biofilm. Once the thick biofilm is formed, it liberates some toxic substance and ultimately it kills the fishes. So we have some eco-friendly approach. So we grow the algae, we treat the cyanobacteria, particularly spirulina, as a feed in the aquaculture industries, aquaculture practice, or aquaculture ponds. So whenever the quorum sensing signals liberated from the pathogenic bacteria, in sensing the quorum sensing signals, the spirulina, the cyanobacteria, liberates the anti-quorum sensing compounds. So the anti-quorum sensing compounds binds with the quorum sensing signals that are the compounds, and it forms a complex. So the quorum sensing signals, as a result, it will not reach the similar kind of pathogenic bacteria in the environment. So by this way, we stop the biofilm development. So if there is no biofilm, there will not be any harmness in the fishes. Otherwise, usually the aquaculture is dumping the antibiotics to kill the fishes, to kill the Vibrio species. But knowingly or unknowingly, we are also consuming the antibiotic-fed fishes, and it is biomagnified in our body. It causes some side effects. Whereas our approach using the cyanobacteria, it prevents the biofilm formation through the anti-quorum sensing or quorum quenching compounds. This interesting result we published in Good Impact Factor Journal also. In one paper we published in the Journal of Applied Phycology, impact of 2.5, with you can see the confocal view of the inhibition of the biofilms. Another paper we published in biofouling. The person involved, he is also my PhD student. He completed recently, and he published the paper in the biofouling and microbial pathogenesis. And now he is in USA as a postdoctoral researcher working similar kind of work in the laboratory. And coming to the nanoparticle, as I said, the nanoparticles are very important in recent development. It is act as a very good carrier for drug delivery and so on. The specialty of the nanoparticle, as I said, whatever size, whatever shape of nanoparticle you want, you just select the microalgae and you can get the desired size of the nanoparticle. For example, so initially we want to develop the golden nanoparticles by using the green algae cholesterol and another blue-green algae formidium. So we treated the gold chloride, and we found that the cholesterol species producing spherical type of nanoparticle, and formidium producing the triangular and hexagonal type of nanoparticles. So again, we published this paper in colloids and surface B interfaces, which is now it is 1.4, 4.4 impact factor. The person involved is Dr. Mubarak Ali. He is in Crescent Institute. And in addition, we select some gelidium, the red algae, using the gold nanoparticle, and we can found that you can get some cubical type of nanoparticles. From that paper also we published in microbial pathogenesis with the 2 impact factor journal. And as a result, whatever size, whatever shape you want, you select a different type of microalgae and cyanobacteria, we get the desired type and shape of the nanoparticles. And we published quite a number of papers published using the nanoparticle synthesis by materials letters and colloid surface B interfaces and annals of microbiology and so on. A good number of papers we published. And coming to the bioenergy, biodiesel. So biodiesel is a very simplified process. We need only the algal oil. So if algal oil is there, we trans-esterificate the algal oil, and we are getting the fatty acid methyl ester and the glycerol is the side product. So this is the triglycerides. That is the algal lipids, and we treat with the methanol or NaOH. We can get the fatty acid methyl esters. The fatty acid methyl esters are nothing but the biodiesel. The glycerol is a side product. And this is, I got it from the literature. In comparison with the plant oils using different sources. And here this slide shows that we are getting the maximum of around 6,000 liters per year per hectare area using the palm. Whereas the same 1 liter per year per hectare area, if you grow the algae, we can get more than 1 lakh liter of algal oil. So this is possible because if you are constructing the 7 raceway ponds, if you are inoculating the algae in the first raceway pond, 7th day you can harvest. If you inoculate the algae in the second raceway pond, 8th day you can harvest. Or third raceway, you can harvest in 9th day. Likewise, first 1 week you are not going to harvest any product, and after 7th day, throughout the year, every day you can harvest the biomass. So this is called sustained biomass production is possible using the microalgae. It is scientifically proved. And this is one species we isolated, which a high oil content, algal oil content. From the vegetative cells, the oil is oozing out from the vegetative cells. For example, if this is the videograph, you can see this oil is drop by drop. It is coming out from the cells. So this is very potential algae called Botricoccus brownii. This particular species having this capacity. So this kind of algae we can utilize for the biodiesel production. And this is the one another student, Dr. Praveen Kumar. Now he is in assistant professor in Denmark. He is my also PhD student. He initially worked on the biodiesel production, giving different kind of stresses to enhance the lipid accumulation. He published this one paper in Biomass and Bioenergy. So by seeing this paper, he received an invitation from the Korean Energy Research Institute and joined as a research scholar, research associate. Subsequently, he moved to Denmark as an assistant professor from Korea. And thanks to the Department of Biotechnology, we got one Indo-UK project of nearly 1.5 crores. From this, we purchased one mobile taxonomy laboratory. So by using this laboratory, we are going to the remote places where there is no pollution. So because wherever the water bodies are there, it is highly polluted. Only pollution tolerant microalgae and cyanobacteria is in bloom form, whereas other algae we cannot see it because it is not tolerated in the polluted environment. For this, to get the wealth of biodiversity, we are going to remote places where there is no pollution. We are isolating, purifying the compounds from the new areas, unpolluted areas. So this is the way of developing the biodiesel from the scratch. So initially, this is the chlorella organism. We are doing some sticking method for purification. We are taking one small colonies, one small dot, and transfer to 10 ml tube, 10 ml tube with the culture medium. And after 2 or 3 days, we got some good growth. After this, we transfer the 10 ml culture established in the 10 ml tube, transfer to 250 ml flask, and we wait for 1 or 2 days, and then we are getting the huge biomass. And from this, we transfer to 2.5 liter Hofkin flasks. From single colony, now we could get the biomass in 2.5 liter flasks. And this is the one, the sodium arginate beads for culture maintenance or the preservation of the cultures. And from this 2.5 liter, we transfer the biomass to 25 liter water can. From this 25 liter water can, we are using, 125 liter tubs. So I don't know who is some barbarian activities are there. Some, some red markings are there. Nivas, Dr. Nivas. Dr. Nivas is there?
Yes, sir. Yes, sir. He is here.
So why this is happening? So I'm not, I'm sharing my valuable time with you. Some
Yes, sir.
ambition people are doing this kind of work.
Yes, sir, I can't able to find among the 100 members, sir. I think some people using in mobile. I mistakenly they are doing while we are connecting the mobile. First thing is it's showing the paintbrush. It might be they are using the Android mobile without their knowledge. They are doing the. I think so, sir. In the morning session also one strike was there.
Okay.
We thought that it might be mistakenly, but again it's showing. I'm addressing it, sir.
Okay.
Kindly hold this, please, sir.
Okay.
Please.
So this is from the 125 liter biomass, we transfer to 5,000 liter raceway pond. So gradually we enhancing the biomass for the biodiesel production. From this 5,000 liter raceway inoculum, we transfer to nearby 35,000 liter raceway pond. This is the biggest raceway pond in any university system in our country. This is the biggest raceway pond we established. You can see this algae growing in the 35,000 liter water. This media. Here we are using only the water, and we are using the effluent sewage water as a nutrient sources. So here there are three-phased potentials. One is we are consuming the wastewater and bioremediate the wastewater, and we use this water as a nutrient source. And because of the growth, if you grow 1 gram of algae dry weight, it already fixes 1.83 gram of the carbon dioxide from the atmosphere. So it is a very good sequestration capability. In addition to this, we are developing the biodiesel, glycerol, and biobutanol, everything from this developed microalgae. And this is how you can see this the raceway pond, how it works with the paddle wheel. So agitation is very, very important because all the cells should exposed to sunlight, then only it grows, multiplies very fastly. Okay, so every day our students are maintaining the water loss, water level, and then pH, and then what are the nutrient loss. Every day they are maintaining the other parameters, so that every day we are harvesting the microalgal biomass through this ratio pond system. And this is the structure of the detailed structure of the ratio pond. You can see this in the Google map also, our ratio pond in the Google map. And this is the one, the efficiency of CO2 utilization by the biomass. So here every month, this is not the optimized harvesting of the biomass, because our students, whenever they're free, they go and harvest the biomass. So it varies day by day or month by month. So here throughout the year we harvested. This is because of the potentials and the capability of our students harvesting. So approximately 1 year we harvested 1.04 kilos of dry weight, kilos of algal powder. And if it is converted to the CO2 sequestration into 1.83, so it already fixes 191 kilograms of CO2 from the atmosphere. So this proves that it is a very efficient CO2 fixer. And you can see this algal bloom. So I applied one project to DBT, how to develop the algal bloom in the raceway pond system. That is artificial induction of algal bloom in the raceway pond system. After I received nearly 50 lakhs rupees for this project, and we are successful in generating the algal bloom in our raceway pond system. So this is the algal bloom we developed during May 4, 2016, I just sent this photograph to my teacher, my PhD mentor, who was at that time in the US. So he was very happy to see this development, the establishment of algal bloom. He said, so I am happy, but I don't believe this photograph, because you have to repeat it repeatedly to its at least 5 times. The 5 times, if you are getting the algal bloom, then only I believe. So likewise, this is in May 2016, and again we harvest everything and inoculate freshly. And this is August 9, 2016. Again, we are getting the similar kind of algal bloom. So from this, we are happy that we developed the algal bloom in the ratio pond system. Now this process is under patenting. And this is how we are harvesting using the plankton net, because we have to minimize the cost of production. So for the harvesting and drying, if you go for the instrumentation, we need the continuous centrifuge and electrical dryer. For this biomass, we need nearly 40 lakhs rupees for developing this harvesting and drying. But it is a very costlier one. If you use this instrument, the algal oil, that is biodiesel, the cost benefit ratio is nothing. So biodiesel cost will be, if you are using the instrumentation, will be more than 500 rupees. So this is not commercially feasible. So that's why we use the manual techniques by direct harvesting in plankton net. And for development of the algal biodiesel, we have to develop two different technologies. One is the rapid biomass production. So once the biomass is formed, then give some kind of stress to induce the lipid production. So lipid is the ultimate aim for biodiesel production. So we use several stress factors like pH change, nutrient deficiency, and inoculating the wastewater or fresh water or marine water. So different kind of stress we give, so we could enhance 4 to 5%. So originally the chlorella having 18 to 19% of the lipid. So if you are giving any kind of stress, 22 to 24% of the lipid we can get it. So this is not sufficient. So for this, we installed, this is the first of its kind, we installed the pulse magnetic field system with 10 channel pulse magnetic field system. So first day we give some very minimal frequency of less than 100 pulse magnetic field. So in the evening, all the algae is tend to die. So it is almost in death phase, reaching the death phase. Then we remove the 10 channel. Instead of 10 channel, we installed a single channel pulse magnetic field. To our surprise, it is a world record. To our surprise, from 20% of the lipid, by treating with the pulse magnetic field, we are getting more than 55% of the lipid. So this is the highest quantity. It is no one achieved so far. And this is the data we got it. And we use the different frequencies, single channel with different magnetic flux, 500, 600, 700, 800, 900. Among these 700, we are getting the 55.2% of the lipid, enhanced lipid, that too on the second day. So this is a very excellent technology. Now this paper we submitted, it is accepted in Chemosphere journal. It is a highly impact factor journal. It is accepted for publication. And initially, if you are collecting the algae, algal biomass using the plankton net, it is in semi-solid content, so it is very liquidified. So in order to develop the noodles-like thing for drying, so we have to get rid of water. So I asked my students to develop a plankton net bag and pour the biomass and squeeze it. So as a result, we are getting some thick consistency. So this consistency required for the making the noodles. So first day, our students are very happily squeeze the bags, plankton net bags. And second day, they feel that all the finger joints are swelled. It is very painful. So then we thought that this technology is not possible or feasible. So we have to go for alternate technology. We think differently, and we, develop a first patent. So instead of spending 40 lakhs rupees for harvesting and drying, just spending nearly 1,600 rupees, we developed another technology, and we got a first patent on this technology. This is the particular technology, that is frame moves out technology for algal harvesting and drying. You can see the uniform thickened flakes we are getting. This is the technology we developed, and I'm not going to talk about anything about the technology. It is filed in the patent, but we are waiting for the grant of the patent. And this is the patent in the patent catalog. Our patent is there, and once we get the flakes, so we do the powder, the small mill to get the powder. And this is the overall view of our technology. So algal biomass with stress condition, we extract the lipids. From these lipids, by transesterification, we are getting the biodiesel, and the glycerol is the side product. So remaining waste biomass after extracting the lipid, we go for the fermentation, inoculate the Clostridium acetobutylicum, we are getting the butanol, Zymomonas, we are getting the ethanol. So as a result, though, no wastage of the product. So this paper we published in a good impact factor journal called Renewable Energy, and you can see this our vice chancellor lighting the lamp, which is a very closed lamp for inauguration of one smart classroom. The lamp is closed because it is filled with microalgal biodiesel. And another threat that we are facing in front of us is the ESBL producing pathogenic bacteria. That is extended beta lactamase, extended spectrum beta lactamase producing pathogenic bacteria. If anyone affected with this bacteria, inhibit this particular bacteria, whatever antibiotic we are treating, they degrade the antibiotic, it consumes the antibiotic as a nutrient source. It grows very fastly. Now we have to go for alternate antibacterial, antibiotic compound for inhibiting the ESBL producing bacteria. One of my students, he worked on this anti-ESBL producing compounds from the cyanobacteria. His name is Parvez Ahmed. So identified the oscillatory acuminator species, and he went up to the molecular level, chemical compound level, that is MGDC palmitolide. He identified, and he published this paper in Nature Group Journal, that is Journal of Antibiotics. By just publishing this paper, he received an invitation from the University of Texas Health Sciences. Now he is a research associate there. Per month he is earning, very proudly says that per month he is earning nearly 3 lakhs rupees without any income tax. So I am very proud to say that the student is very from the poor background. Now he is in the top stage of research. And another student from Assam, Dr. Dhartiri Bora, she worked on the biolubricant potentials of the extrapolysaccharide cyanobacteria. It is also published in Applied Microbiology and Biotechnology, a good impact factor journal. And this is not my work, just to show the importance of the cyanobacteria. If anyone produce the monoclonal antibody using the mammalian cell culture, so we have to spend nearly $150 per gram. But if you're instead of mammalian cell culture, if you are using any algae, the cost is only $0.5, that is 50 cents. So this is according to the Mayfield et al. He published during 2003. You know the potentials of the cyanobacteria. And final part is the cyanobacterial pigments. As I said, it is very attractive, colorful pigments. We can get it from the cyanobacteria. And you can see this cake is decorated with the phycocyanin pigment. This is the marine cyanobacterial phycocyanin. This is Formidium valerianum species. You see the different shade of the blue color. And one of my PhD student, now she is assistant professor in Jamal Mohammed College, Dr. Rihana. Her PhD work is on pigment production, so phycocyanin pigment production from the freshwater cyanobacterium Oscillatoria geitleriana. You can see the different shades of the blue color. And yes, we can inoculate the mix in the ice cream as well as the milk. Instead of rose milk, we can consume it as a blue milk because it is a plant protein. There is no side effect, which has the high nutritive value. And recently we isolated one red colored organism. It is a blue-green algae, but it has the high phycoerythrin content. We extract the phycoerythrin content also. So here you can see this cake is decorated in both phycocyanin, blue color, and purple color phycoerythrin. Cake is decorated, and we are mixed the phycocyanin and phycoerythrin in 1:1. we are getting the lavender color. So by this way, now we have a plan to generate almost all the colors in the, just like a inkjet printer. Only 4 colors are there. But whatever color you want, by proportionate mixing, we are getting the different colors. Now we have different colored cyanobacteria also, the dark brown color, yellow color, different cyanobacteria we isolated. Now we are about to establish a technology that is natural color and technology. Just like going to the paint shop, and if you ask some shades, they have one proportional mixing with algorithm, we are getting the desired color. Likewise, we are in a plan to develop, generate almost all the colors, totally, completely eradicate all the synthetic food colors, which are in turn, long term, it is carcinogenic. So my boss, Dr. G. Subramanian, during his period, he developed some technology and transferred the technologies like phycosin pigment production, beta lactamase enzyme production, aquaculture feed production. Also, it transferred, and he collaborated with Dr. S.P. Tyagarajan, who was the virology professor during those days. And we have a collaboration. And the formidable multi extract we supply to Dr. S.P. Tyagarajan's laboratory, SARS laboratory, and he found that it has the anti-AIDS virus activity by inhibiting the reverse transcriptase enzyme. So now this technology is also transferred to one company. And we made some patents, 2 Indian patents and 2 US patents from our laboratory. These are the persons responsible for the patents. And thanks to DBT, they sanctioned one a national repository for freshwater microalgae and cyanobacteria. And this is the half of the building we constructed. This is the proposed building. Now we are waiting for this fund. I think this month we are receiving the fund. We will complete the building structure, total national facility. So what happened?
So now it's clear, sir.
It is almost over. Okay. So recently, 2018 September, Dr. Sangeeta Kasturi, she is the joint director from DBT. She declared open the building in front of our Vice Chancellor and Professor Anand and our dignitaries. So this is, the facility, infrastructure facility equipped in the national repository, culture collection centers, and instrumentation and pilot scale facility. Almost all kind of microscopes we equipped in our national center, and we have the website also, NRMCF website. You can get all the details, even the isolation, how to isolate the cyanobacteria, methodologies, microphoto, everything is available in the website. You can easily access. And we pasted the distinguished algologists and cyanobacterials of the world to just show their contribution to the young generations, young scientists. And he is my professor, Dr. G. Subramaniam. Because of him, I developed this kind of technologies, and I'm talking in front of you. And this is the last 4 slides I like to purposely show you all. When I was in DBT Overseas Associate Fellowship, I entered the US during the spring season. At the time, the tree, I thought that it's a flower, but my guide says that it is the leaf buds. It is in a very attractive purple color. So this is in the spring season. So I sit under the tree and took the photograph. And the second one, the same tree, in the summer season, you see the photosynthetic activity, it is very luxuriant, and all the developmental process in the tree. I sit under the microscope under this tree and took the photograph. This is in the summer season. The other season, again the same tree, I am sitting under the tree. In the summer season, the fall, this is called the fall season because of the temperature drop. So it is a stress. So all the leaves are fallen. This is a fall season, and the same tree in the winter season. You can see everything is covered by the snow. Again, I sit under this tree, took the photograph to show the different seasons. So why I am showing these 4 slides means, we are really blessed to live in the tropical countries in India. So there is no seasonal variations are there. If you develop open tank cultivation of algae in any open tank cultivation, it is possible only with the tropical countries like India, not the Western countries. So that's why All the multinationals are putting their companies in our country because of the tropical nature, and also we are one among the 14 mega biodiversity of the country, and we have a very excellent flora and fauna. Only thing is we need the trained and focused human resource to exploit not only the sign of but exploit all the biodiversity for our benefit and made patent, and then then only we will say that we are in a developed country stage. So now it is up to the young students to develop themselves. So with this brief my note, I thank each and every one of you for very passionately listening my talk, and also I thank the organizer, Mr. Niyaz Ahmed, and the conveners, Dr. K.R. Sivana and Dr. DJ Bhakiraj, for their support in organizing this kind of the Science Academies workshop. Thank you, one and all. If you have any doubts, you can feel free to ask.
Yes, sir. Thank you, sir. Thank you so much for your nice presentation. Some participants asking the questions. Shall I ask, sir?
Yes, sure.
So the first question, Kishore from Maharashtra. Which method are used for the extraction and isolations of algal compounds?
Actually, there are so many methods are available in the literature. For like extraction of lipid, we are using the Bolt's method, and for extraction of the the pigments, we are using only the distilled water. There is no solvent we are using. Likewise, each and every component, there are specific methods are there. So we have to identify the suitable methods in the literature, and also the type of organism we are selecting for the extraction of the compounds. For example, if you isolate the DNA, for example, there is one common method is there, but sometimes because of the thickness of the cell wall, that method will not be used for our algae because the very thick cell wall. So we have to add more quantity of the lysozyme or some sonication or some crushing. So different techniques you have to follow. So it depends on what type of organism we are using. We have to standardize the methodology. Then only we will success.
Okay, thank you, sir. So next question from Ramya. If there any microalgae which produce enzyme to bioremediate heavy metal pollution?
Yes. Because of the secondary metabolites only, the toxicity will be gone. The degradation of the pigments, colorant, and initially the effluent are dark in color because of the secondary metabolites, and also the oxygen. So initially the effluent is not having any oxygen. It has the high BOD, COD level. The anaerobic bacteria will be there. It is toxic. Whereas if you are growing the algae, it liberates the oxygen during the photosynthesis, so it automatically drop the BOD and COD level. So oxygen is there. All kind of positive points will be there. So in addition, the organism liberating some secondary metabolites, even the pH change. If it is acidic effluent, if you use the algae, it by liberating the secondary metabolites, we can get the neutral environment, neutral water, instead of the acidic or alkaline. Likewise, it is all in all. So if you're using the algae, definitely you're getting the 100% success in any product or process.
Okay, sir. Sir, one more question. She asked, what is the role of microalgae in bioremediations of heavy metal?
So there is a adsorption is there. There are several processes. One is absorption, and bioconversion are there. So sometimes adsorption means because of the mucilaginous sheath, it is attracted in the mucilaginous sheath. So it is called adsorption. Sometimes bioconversion, as I said, the anthracene and naphthalene, the compound structure will be varied, bioconverted. Even the chromium 6 converted to chromium 3 and so on, from toxic form to non-toxic form. So it is possible because of the secondary metabolites.
Okay, sir. Sir, one more question in Zoom. So the how to determine the nutraceutical values of algae?
So by animal experiment, we can determine this one. So and also the toxicity tests, everything in some standard laboratories are there. They have the institutional ethical committee and the biosafety committee, animal ethical committee, and laboratories are there. If you are giving our product, they will do it, and they analyze the toxicity and what are the benefits, everything they will deliver to you.
Okay, sir. Sir, one more question, sir. What is the best microalgae strains for biodiesel production?
And we have different type of algae, and now there is a concept that the consortium is better. So now whatever I showed is only the chlorella, because the chlorella is a very robust organism. So it tolerate all the different climates, even heavy sunlight or very dim sunlight or any cooling condition. This organism is very robust and grows very faster. So we identify the organism which are growing very faster, then only we can select this, and we can manipulate the media composition and condition for enhancing the lipid productivity. I think I answered that question.
Yes, sir. Yes, sir. Thank you, sir. So now I call Dr. I. Nia Sadamas, the coordinator, to deliver.
Oh, thank you, Dr. Pongodai, for your moderator. I would like to thank our resource person, Dr. N. Tajuddin, Professor and Head, Department of Microbiology, Dean of Faculty, Dean of the... Ma'am, can you hear me, ma'am?
Sir, yes, sir. Yes,
Sir,
yes.
Dr. Riyaz Ahmad, I am not presently head BTA because it is a rotation. I am a professor and a chair, and my deanship is also over. So
Okay, fine.
I was the dean for around 4 years. Now another person, because in our university everything is rotation.
Okay, fine, sir. Fine. Utilizing all your expertise, that's why they are doing cyclic process. Okay, I'm getting you, sir. Getting you, sir. With this renowned personality, we are interacted with him. I would like to thank him for his valuable presence. He gave the very good outline behind the microalgae, biodiversity to biotechnology with entrepreneurship-oriented lectures. Each and every slides give the very good enthusiastic to us, our students, as well as the students from other participants. Thanks for his very good lecture. Sir, there is one question from the YouTube live streaming, sir.
Okay.
Which is the purification process for the isolated algae? What kind of purification process we have the ideal method?
Purification. Actually, we are using the conventional method like a streak plate and pore plate and petri plate. Different methods are there. And usually the algae is symbiotically associated with the bacteria. So whatever we are saying, it is a uni-algal culture. If you want to make the oxenic cultures, we have to use the different type of antibiotic, different combination of antibiotic to get rid of the bacterial contamination with the algae. So for the uni-algae, we use some streak plate or pore plate or spread plate method we are getting. And for this, we have to use the different combination of antibiotics. In this connection, I like to share with you that we made the second patent. Instead of using the antibiotics for killing the bacteria, in our second patent, we use the ozone gas. So we spark the ozone gas in the culture medium in particular proportion and concentration. The bacteria only eliminated, and only we are getting the cyanobacterial, that is unialgal growth. So that is our second patent we made it. So without using any antibiotic, for antibiotic treatment, we have to wait for getting the oxenic cultures. Whereas in using the ozone gas, so within 1 week, we can get the oxenic cultures. So this is our second patent we made.
Oh, thank you, sir. Thank you, sir, because we have 100 members in the Zoom link and around 150 members in the YouTube live stream. That's why we can't able to find some person anonymously putting some
that is something irritating
yes,
us.
Yes, sir. Yes, sir. That's exactly I have instructed through the group chat. Then only some people has erased it. Those who did the mistake, they only can able to write it. That's it. Before your thank you slide only they are address the issue. Similarly, in the morning session also some person, because we can't able to find him. I think that he without his knowledge or those member who's using in the mobile phone, they did this because they are scrolling down. Sorry for the inconvenience, sir. On behalf of the organized committee, I would like to your grievances. Thank you for a valuable present, sir. Thanks a lot, sir.
Thank you. Thank you.
Happy discussion with you. Thanks a lot, sir.
Thank you. Thank you. So I'm leaving
you, sir.
this.
Thank you, sir.
Thank you.
Thank you, sir. Participants are requested to kindly be in line. Dr. Bharati Salimat is going to join. She is supposed to join within 2 to 3 minutes. Dr. M. Fernandez Turai, Assistant Professor of Department of Biochemistry, is going to moderate the session. Over to Dr. Fernandez.
Dear participant, kindly be in the line. Just now I spoken with Dr. Bharati Salimat. She is supposed to join within 5 minutes. Some technical issues are there. She is supposed to join within 10 minutes. Hope we can join with Dr. Salimat. Then if time permits, we will complete the session around 5 o'clock itself. Kindly be in the line. Yes, ma'am. Welcome, ma'am. Good evening, ma'am. Ma'am, your mic is mute condition. Kindly unmute yourself, ma'am. Professor Bharati Salimat. Kindly, ma'am, kindly unmute yourself.
I have unmuted.
Yes, ma'am. Got you, ma'am. Welcome, ma'am. Welcome to the virtual conference.
Thank you.
Yeah. Now I call Dr. Fernandez Turai to take over the session.
Okay.
So very good afternoon.
Audio is very poor.
1 second, ma'am. Dr. Fernandez, we are hearing you. Kindly raise your voice.
Wait.
Can you hear me?
Yes, ma'am. Yes, ma'am.
Fine.
One name share. Good afternoon, ma'am.
Good afternoon. Yes, I can.
Thank you, ma'am. Yes.
Yes, doctor, you can go ahead, please.
So very good afternoon. In the last session, I welcome you all. So with this, I feel proud and my privilege to introduce about Professor Bharat P. Salimat. A professor has done MSc in biochemistry from University of Mysore in 1978-1980 and PhD in biochemistry from Albert Ludwigs University, Freiburg, Germany in 1983. And Doctor was awarded postdoc fellowship by Albert Heritage Foundation for Medical Research in 1983 to work at University of Calgary, Canada till 1985. Dr. joined as a scientist of Pool Officer of CFTRI Mysore in 1985 before taking an academic position in the Department of Biochemistry, University of Mysore in 1987. Presently, Professor is working as a Professor of Biotechnology in Department of Biotechnology, University of Mysore, Manasagaranati, Mysore. She has more than 3 decades of research experience. Her field of research is molecular oncology with an emphasis on tumor angiogenesis. Her research has been funded by various government funding agencies. She has successfully guided 27 students in research work leading to a PhD degree and has published more than 80 research papers in peer-reviewed international journal with high impact factor. Professor is keenly involved in scientific activity of various science and technology division of the country. He is a visiting scientist to Tumor Biology Center, Freiburg, Germany with Christian Work University of South California, USA. The Government of India, DBT, Associateship and Georgia Technological University, Atlanta, USA with the DBT Cresta Fellowship. Professor has been awarded with the Virendra B. Kamath Awarded in 1995, Young Scientist Awarded in 1996, and Dr. Kalpana Chawla Awarded in 2003. And Corona Tech Transfer Program in 2012 for her exceptional contribution to cancer research and biotechnology. Dr. Bar Salamat has been awarded in 2020 with the prestigious BSR Faculty Fellowship by UGC. Currently, she is a UGC BSR Faculty Fellow at the Department of Biotechnology, University of Mysore. Dr. Bharat Salamat is an academician, entrepreneur. She has inducted biotech companion Sonarova Biotech and Malmed Research Foundation in Mysuru with particular emphasis on empowering skill development and hands-on training workshop in life science for students, teachers, and research scholars from all over India. So, this I welcome Professor to take the session. Participants.
Thank you very much for your kind introduction. I will share my slides now so that we can directly
Yes, ma'am.
go to the lecture. Visible?
Yes, ma'am, visible.
Okay, audible and visible.
Yes,
So
ma'am,
we
audible and
are
visible.
the right platform. I hope all lectures have gone very well for you from morning. And thank you for giving me this opportunity to talk to all of you and also for those people who have logged in. Today I will cover my lecture as 2 aspects. One is on molecular biology of cancer. The other one will continue with this to introduce to you all some aspects, examples of how the understanding of molecular biology of cancer has helped mankind to design drugs for treatment of cancer. Cancer, you all know, is a very devastating disease. And as soon as a person hears, he or she, that they have cancer, they get scared about it. The only reason why they get scared about it is because of the process called metastasis. Screen sharing has stopped or what? Can you see the screen?
Now,
Can
now,
you see the
now we
screen?
can see.
Okay,
Yes, ma'am. Yes, ma'am. It's now visible.
okay, fine. So basically in molecular biology of cancer, what we are going to study is the characteristic features of cancer cells, and several names are given for cancer, and we will discuss that. And also I like to convince you that cancer is multiple genetic defect. It is as a consequence of genetic defects in cells. So we like to approach this disease with molecular genetics, and we will see what are the genes involved and how they are manipulated inside the cell, either by environment or by chemicals or by viruses. Some examples will be given for that. And we will also see great number of signaling pathways involved in process of cancer, and discuss some things about the treatment. Why it is important to study cancer is about 1 in 4 of us will get cancer and 1 in 5 of us will die of cancer. This is not the exact number, but I like to impress upon you that this is a disease where still many research events have to take place. This is a disease where many new drugs have to be identified. But over the years, people have understood the disease very well. And to that extent, they have designed the drugs. But there is still a lot to understand about this disease at molecular level. And because many people are dying in spite of taking drugs, right now, not all people who get cancer will survive. So you see, even in 1880s, a painter called Van Gogh, he did not know anything about cancer, right? But a beautiful painting depicting smoking. So already, we are taught, like we kept thinking, or we keep thinking, even in age-old days, where smoking existed, people must have died with cancers. So right now you can recognize that smoking is injurious to health and it can lead to cancer. Likewise, there are many types of cancers like prostate cancer, breast cancer, lung cancer, both in men and women, colorectal cancer, right? Lymphoma, bladder cancer, melanoma, cancer of the kidney, leukemia, pancreatic cancer, ovarian cancer, brain cancer, cancer of the soft tissue. You name any organ, skin cancer, right? You name any organ, there is cancer. So all this, we start wondering, how many deaths are there and what percentage of population will get, why they get cancer, and so on. So if we want to understand the basic properties of cancer cells, the most important aspect is that cancer cells grow in an uncontrolled manner. They grow at usual rate, but then they keep on growing. When other normal cells will stop growing, cancer cells will never stop growing. So what do? They ignore the stopping signals. So they grow even without adding any signal. When we have to grow, we have to eat food, get nutrient, oxygen. Everything is possible because we take the signals from food or environment, right? And we grow. Normally we grow. But in cancer cells, when there is cancer, there is uncontrolled growth. Even without taking food, even without taking signals, even without having growth factors, right? Supplying growth factors from outside is not required for growth of cancer. So they grow in an uncontrolled manner. If cancer grows in one place and if it is uncontrolled, it grows and becomes a lump, but nothing will happen. It will stay there in one place. But what if it travels to other places in the body? Then it is called as being invasive. It moves from a primary site, wherever the cancer occurred, to a distant site in the body, goes and lodges there, and again a secondary tumor will be formed in a different organ in the body. So cancer, as long as it is in one place, fine, one can get it removed by surgery. But when it starts traveling to other parts of the body, when it becomes invasive, then only the problem occurs. And there are chances that a person may not survive. So we will discuss about these aspects in next few slides. Here you can see a pictorial representation of how well-behaved cells will grow, and they stop growing in this dish. When cells have covered the whole of the dish, when cells have been touching to one another, they respect the boundary of the neighboring cells, and therefore they stop growing. They don't have place. This is normal growth, and this particular layer is called a monolayer. They form a single layer on a substratum. However, in cancer cells, you don't need such widespread substratum. On the substratum, the cells have grown. They respect the boundaries. You know, this is a microscopic view of the same. But cancer cells, you see, they don't respect the boundary. They heap on one another. They keep growing. In spite of the dish being covered, they overgrow on one another. They have formed clumps, and these are called as foci. This is how the picture looks under microscope, and see how different it is compared to the previous one, where there was contact inhibition and the normal cells had stopped growing, but cancerous cells, they heap on one another, they form foci, and they keep on growing. And you can see here a normal cell in this blue dotted line at the bottom of this graph, right? They don't grow in the absence of growth factors. If you don't provide the growth factors in a culture dish, they cannot grow. Normal cells cannot grow. You have to add some serum growth factors in order to take off a growth curve. This is in case of normal cells. But what about cancer cells? Even in the absence of growth factor, they grow very well, right? And suppose you add growth factor, further growth will be there. They respond, but you know, even in the absence of growth factor, they can grow. So that has a meaning, because cancer cells per se by themselves can produce lot of growth factors. So they're self-sustained, they can grow in an unlimited manner, and they don't respect the boundary of the neighboring cells, and they have an uncontrolled growth. There are several names given to cancer. People call them as tumor or neoplasm, that is the new growth. And these are clone of cells with uncontrolled growth. I did mention they are contained in one place or they are spreading type. If they are contained in one place, they are called as benign tumors. If they are the spreading sort, they are called as the malignant tumors. And they are classified by tissue of origin, where they occur. If they occur in epithelial tissue, it is called carcinoma. If they occur in mesodermal, or if they occur in tissues of mesodermal origin, like muscles, connective tissue, vascular tissue, they are called as sarcoma. They are called leukemia or lymphoma if the cells in the blood, that is the hemopoietic cells, become cancerous. What is the current view of cancer? Cancer arises through a multi-stage process that involves inherited and somatic mutations of cellular genes. So this is the current view about cancer, how it arises. So it is a multi-stage process, and it involves inherited and somatic mutations of several cellular genes. Many mutations in several distinct genes are required for a normal cell to become a cancerous cell. The progressive accumulation of these multiple hits will correlate with the age-related incidence of cancer. It doesn't happen overnight. So you can see this is a age-related incidence. There will be progressive accumulation of multiple mutations. As a consequence, a person will get cancer. And we say cancers are clonal, means they arise from a single cell. The DNA in a single cell gets mutated, and many other cells which come from or which multiply from this mutated cell will cause a tumor or a cancer, right? So all cells in a tumor originate from a single ancestral cell which was mutated. But not all cells in a tumor have the same genotype because the cells are genetically unstable. Cancer cells are genetically unstable. Because of this unstableness of cancer cells, it gives rise to selection. The variation you see between the cancer cells will give rise to selection. Clonal selection of variant progeny with the most robust growth property will play a major contributing role. So, if there is a variant and if it has robust growth property and clonally if it is selected, certainly it will lead to very rigorous type of cancer. What does this clonal selection of cancer look like? You can see here, these are all the normal cells, and in a nucleus here, in one cell, you can see some mutation. It's only a pictorial representation. As a consequence of the cell division, you can see both the cells have mutation, and number of mutations are increasing, further increasing, and a lot of mutation and lot of cell division has taken place to give rise to a tumor. And these are different variants over time. You can see they are the variants over time. And a clone which has a robust growth property will succeed in becoming a cancer. So we understood that the origin of cancer cells is dependent on the clonal selection, but it is highly mutable. It will get mutated, right? One best example for that is CML, that is chronic myelogenous leukemia. We will study about this a bit later, where major changes in the chromosomes will be there. So more than one defect is involved when a cancer occurs. In a lifetime, 10 to the power of 16 cell divisions may occur, and mutation rate is very high. But whenever there is mutation, we have the good part in our body that the mutations can be reversed, and there are checkpoints and there are repair enzymes which can take care of the mutation. If all these events are bypassed, it will lead to cancer. So you can see here with age, this example of colon cancer, where with age, the rate of cancer increases. So whatever molecular ideas one has, or right now people know, it agrees with this aspect that frequency of cancer per year goes up with the age because mutations will go on accumulating with age. So this process of progression of a particular tumor, say for example, cervical carcinoma, the cells can be in a precancerous stage. Though the cells are undifferentiated, they are in a precancerous stage. Some may be immortal, but still they are not cancerous. Only a fraction of precancerous cells will become cancerous. Thus, the changes are typically sequential rather than having to be simultaneous. So sequential mutations occur in order to define a phenotype of a cancer. Here you can see the cells can revert to a less differentiated stage, and carcinoembryonic antigen is expressed in embryos, and such antigen is also expressed in cancers. The more the number of mutations, with time, more will be the chances of occurring of cancer. So with this molecular evidence, it is like we are very sure that cancer is occurring because of multiple mutations, and with advent of cloned oncogenes, it is possible to add such genes to normal cells or animals to determine the result. So people have cloned and expressed oncogenes, and with these cloned oncogenes, it is possible to show the results that, you know, normal cells can be transformed to tumor cells. So you add the oncogene is a cancer causing gene. You add to normal cells the oncogene, and the normal cell will turn to be a tumor cell. So single added oncogene in T3 cells, can make them cancerous. Normal cells require two oncogenes, such as, see here in this case, in this purple line, two oncogenes are involved, MYC oncogene and RAS oncogene, to result in transformation of a normal cell to a tumor cell. So genes and cancer are very much related. In this picture, you can see that a normal colon cell, to become a malignant colon cell, has to go through the genes here. The gene losses are because of all mutations. Normal cell will get increased cell growth when a gene called APC gene is lost, and further there is early stage adenoma, DNA loses methyl groups. Further, there is mutation of a gene called the RAS gene, intermediate stage adenoma. Then DCC gene will be lost. Late stage of adenoma occurs. There is loss of P53 gene. The carcinoma is seen, and then it becomes malignant on loss of this PRL3 overexpression. It's a protein related to liver. When it is overexpressed, the normal colon cell will become a malignant cell. This is only further description of the same. There is loss of gene and loss of DNA methyl groups to bring to early stage adenoma, then late stage adenoma, further mutation and further loss of genes will be there. And P53 will be lost, and some genes are overexpressed. As a consequence, there will be malignancy. So what causes all these mutations? It could be because of lifestyle or maybe mutagens. Some cases viruses will cause cancer. Diet can also have an effect. Do the cancer cells differ in their gene expression, and how do they differ? So people have used microarrays to find out what are the different gene expressions occurring in cancer cells. People have identified thousands of gene-specific sequences during this study. People isolate mRNA from the cell and turn it into cDNA and hybridize two sequences in this process of microarray, and lot of information is got on differential gene expression compared to normal cells. When tumor occurs or in cancer cells, one can identify what are the derailment of gene expression. So when a patient goes to a doctor, usually they look for the presence of cancer cells in lymph node, whether patient is negative, where lymph node is concerned, or the patient is positive. So if the patient is positive, that means cancer from the primary site has spread to the lymph node, and further it will spread to other parts of the body. The cells can move in the lymphatics or in the blood vessel to lodge in another place in the body, another tissue in the body. And usually the node positivity indicates it is a poor signature and it is taken as a prognostic indicator. Two basic effects the genes will have on cell growth. They can stimulate the growth of the cell or they can inhibit the excessive growth. Oncogenes are the genes that are normally turned on as part of growth and they will be active. Dominant mutations occur on these genes and also tumor viruses can cause mutations in these genes. As a consequence of inhibition of excessive growth, they are called as the tumor suppressor genes. They suppress the tumor causing genes, they suppress the oncogenes, and they keep the cell division in check. So there are oncogenes and tumor suppressor genes which contribute to the process of cancer. This is a small picture showing you depiction of how the tumor virus genes will act. The virus will produce the proteins. Say the proteins which are produced can inhibit the major tumor suppressor genes. We'll go about this a bit later. This only to show you that viral induced cancers, virus induced cancers, they act through proteins. Like here you can see the protein is binding, and this you can see is the P53 and this is the retinoblastoma. And if a viral protein binds either to retinoblastoma or to P53, viral proteins are E6 protein, E7 protein. They sequester retinoblastoma gene and the P53 gene. Once these are sequestered, cell proliferation will be activated by DNA viruses. Otherwise, when these viral proteins are not there, the retinoblastoma gene and the P53 gene control proliferation very well. They are very much required to regulate cell proliferation. However, because of the virus and the virally induced proteins like E6 and E7, which can sequester, they can bind to retinoblastoma protein and the P53, and they induce gene transcription as a consequence of protein-protein interaction. Most discovered oncogenes were RNA tumor viruses. And homologous genes, similar genes are found in our cells, and these are called proto-oncogenes. Proto-oncogenes and virally derived, proteins, they have similarity. These oncogenes, proto-oncogenes, they code for several proteins. There are several oncogenes coding for several proteins. These proteins which are encoded by oncogenes, they have major role in controlling the cell biology. They can play as signaling molecules as exemplified by RAS, or they can play as cell regulating growth factors, or oncogene encoded growth factor receptors are there, transcription factors are there, anti-apoptosis proteins are there. So proto-oncogene derived proteins in the cell play a major role in cell growth and regulation. This is an example to show only a few of the oncogenes, right? Like ABL gene, BCL2, CDK, cyclin-dependent kinase ERB, new oncogene, MYC oncogene, RAS oncogene, RET oncogene, and so on. And these are the neoplasms. They cause chronic myelogenous leukemia caused by ABL, BCL2, B cell lymphoma, and cyclin D1, the carcinoma of breast. These are only few examples, right? What I have to tell in this slide is that many of the cancers are caused by mutations in the proto-oncogene, which converts a proto-oncogene into an oncogene. A good gene will be converted into a bad gene. And because oncogenes code for major cell regulatory proteins, they bring about the transformation of a normal cell into a tumor cell. So here is the proto-oncogene in this pictorial representation, which contributes to the normal cell growth on one hand. But if a proto-oncogene is predisposed to mutation, mutated proto-oncogene will now become an oncogene. Lot of changes will be there in cell regulation. Lot of growth, loss of growth control will be there. A normal cell will now become a transformed cell. And you know, one single transformed cell is enough to give rise to a tumor. There are different ways to become a bad oncogene. Sorry. So here is an example to show what are the different routes taken by a mutated oncogene to give rise to cellular transformation. A proto-oncogene, when it is expressed, that is transcribed and translated, gives rise to the protein which performs normal cell regulatory functions. However, there are three examples here, A, B, and C, where in example A, there is a deletion mutation. In example B, there is what is called as gene duplication. The same gene will duplicate again and again. And in example C, you can see there is fusion of two different genes. A regulatory sequence will translocate from a distant site and induce the expression of a gene or synthesis of a protein which contains portion of an other gene, right? Because of translocation. So either translocation or gene duplication or deletion mutants all give rise to structures of proteins which are mutated, and these mutated proteins contribute for a cell to transform itself from a normal cell to a tumor cell. So this is the molecular mechanism, an overview, a bird's eye view of how different types of mutations on proto-oncogene can make a proto-oncogene into an oncogene, make a oncogene which can cause cancer. And gene products are the ones which are further responsible for cell regulatory processes which are derailed in cancer. Say, for example, HNPCC, which is located on chromosome 2, 15% of the tumors are being identified with the mutation on this HNPCC oncogene. And this gene is involved in maintenance of the replication process in the cell. KRAS is a very important gene located on chromosome 12, and 50% of the tumors will harbor this mutation on KRAS. So KRAS, an oncogene, it is involved in many signal transduction processes in the cell. Cyclins, various types of cyclins are there, and they are present on various chromosomes. 4% of the tumors harbor mutations in cyclins, and that cyclins are involved in cell cycle regulation. New HER2 gene, an oncogene, 2% of the tumors are identified with mutation of HER2 new gene, and it is located on chromosome 17, and it acts as a growth factor receptor. There are other genes like MYC oncogene, APC oncogene, and P53. Out of these, APC and P53 gene, you can see 70% of the tumors will show mutation of these two tumor suppressor genes, and they regulate the other activity of other genes. They regulate several activities, including gene expression. So far we discussed several of the oncogene products oncogenes and their products, how proto-oncogenes are converted to oncogene upon different types of mutations, and how these mutated oncogenes give rise to proteins, or they express as proteins. And these proteins, they harbor a major role inside the cell. They can either be growth factors acting outside of the cell, or they can act as growth factor receptors. These are all oncogenes, means mutated proto-oncogenes give rise to proteins on expression, which are growth factors, or they can act as signal transducing molecules like SERT, RAS, RAF, right? These are all involved in signal transduction, and they can act as protein kinases. They can activate other protein kinases. And there are several proteins which are expressed, like BCL2, which are involved in controlling the apoptosis. There are cyclins and cyclin dependent kinases. You can see here the fourth category, which control the cell cycle division. And the fifth category is apoptotic controlling genes. Sixth category are the genes which encode for transcription factors. So right from outside of the cell, where these are secreted growth factors, till the nucleus, the whole of signal transduction process in intracellular signaling is heavily controlled by major of these proteins. Very, very important proteins in the cell, the growth factors, the receptors, and signal transducing kinases, apoptosis controlling proteins, and transcription controlling genes. All these are very important for normal living, for normal life of a normal cell. If any one of these proteins go haywire, if PDGF or growth factors are produced in large quantity, if receptors are overexpressed, if kinases get more activity and more of BCL2 is produced, and transcription factors lose control over their activity of transcription, lead to cancer. So these are the major players, 6 groups of major players which can impact upon mutation on the conversion or transformation of a normal cell to a tumor cell. The tumor suppressor genes, they tend to halt unregulated cell growth. Cell cycle control with damage or inappropriate signals. Usually when cell cycle has to occur, there should not be any mutations. There should not be any damage to the cellular DNA. If DNA damage is seen, then the tumor suppressor genes will come into play and they will stop further cell cycle division. And as a consequence, these cells will be rooted to apoptosis. The damaged cell, cellular DNA, if it is not repaired, that cell has to die. If that cell doesn't die, it harbors the mutated genes and it will lead to cancer. Therefore, the tumor suppressor genes, they suppress the division of the cells where the DNA is damaged. And 2 bad copies are required because these are the recessive genes. On both the alleles, mutation of the tumor suppressor genes will kill the function of the tumor suppressor gene. It can no more suppress the cell division cycle, and therefore cells will become mutated. Here is a pictorial representation of such aspect, what I told you. Normally, cell growth will occur and it will stop at a particular signal. However, mutated, if a tumor suppressor gene is mutated, even if one allele is mutated, you can see that loss of the second allele, these copies of tumor suppressor gene on both homologs, these are mutated and it will show loss of growth control. So further mutations will lead to loss of growth control and leading to a transformed cell. This is a nice example to show about the retinoblastoma gene, which again is a tumor suppressor gene besides P53. and involved in hereditary cancer with 90% penetration. And if an individual, a kid, is born with one bad copy, then it's fine, nothing will happen. However, if both copies are mutated, then it is a two hit. So both the copies are mutated, double hit will be there, and it will then affect the growth of the cells, leading normal to tumor formation. And here you can see the retinoblastoma protein is phosphorylated, and it will bind to an elongation factor called as elongation factor 2. When this complex is there, there is gene repression. There is no expression of any gene under the control of this elongation factor. The CDK, cyclin-dependent kinase activation, leads to the phosphorylation of retinoblastoma, and you can see there is dissociation of this phosphorylated protein. Upon phosphorylation, it will leave the E2F, and E2F will induce transcription to give rise to mRNA, which is then translated to the protein. And now the cell cycle will proceed from G1 to S phase, synthetic phase. So if cell cycle has to occur, if it has to move further, the RB protein should not bind to the elongation factor 2. And here E2F can be dissociated because of phosphorylation of the RB protein. The P53, which is called as the guardian of the genome, again is a transcription factor, unstable factor, and it can recognize the DNA damage, and it will induce the checkpoint where P21 is transcribed. This is again a protein which is under the control of P53. It is an inhibitor of cyclin-dependent kinase, and it stops the G1 to S phase transition. But it will activate apoptotic genes as well. That happens only when DNA damage is there. The P53 will become active. It will induce expression of P21, which controls the cell division cycle. And basically, there is activation of apoptosis. Pictorial representation of the same. So here there is the DNA damage, maybe due to radiation or chemical, and P53 level will rise in the cell. P53 overexpression will be there so that it will protect the cell, it will arrest the cell in the G1 phase, and cell division doesn't occur. The cells will undergo apoptosis. If there is repair of the mutated DNA, then the cell will divide further. This arrest will be removed and cell will divide further. This is an example, again, pictorial representation to show that there will be cell death because of mitotic failure. If there is no G1 arrest, right, cell will go into mitotic arrest. And if no G1 arrest is there, when there is no P53, so DNA is damaged, but P53 is mutated, there is no P53, therefore it cannot arrest the cell cycle. The cells will multiply to give rise to tumor cells. Cells cannot go and die. So this, when it happens in the breast cancer genes called as BRCA1 and BRCA2, when mutations on BRCA1 and BRCA2 are repaired, you can see that when there is repair mechanism of the gene, the breast cancer doesn't occur. However, when there is no repair mechanism, the P53 you can see, is mutated and it will give rise to breast cancer. And therefore, this is a checkpoint activation where P53 will induce the expression of P21 and it will arrest the cell cycle as soon as there is damage. So when checkpoint activation is there, P53 will suppress the growth of the tumor. It will induce mutated cells to undergo apoptosis. That is why it is called as a tumor suppressor gene. These are all the chemicals like 5-fluorouracil, etoposide, adriamycin, which are used as for treatment of cancer as chemotherapy. And all these chemotherapeutic agents will operate in the mechanism I just now explained, that the cells will undergo apoptosis, cell death will be there, and tumor cells can be controlled for cell division by chemotherapy. A typical chemotherapy or radiation therapy will take advantage of these subtle differences in process of carcinogenesis, and they can kill a tumor cell. There are new treatments and also very directed treatments, personal treatments, like, person will be thoroughly examined and all the genes which are mutated will be identified, and targeted therapies are available in recent times. This is only an overview. We will cover this aspect a bit later. Likewise, immunotherapy is very popular, and they're made to kill the tumor cells by using antibodies like Herceptin and rituxan, which is an antibody against B cell surface protein, which is used in non-Hodgkin's lymphoma. So immunotherapy is also gaining a lot of importance in treatment of cancer, and Gleevec is a very commonly used drug produced by very good companies, biotech companies all over the world, and it inhibits this ABL gene, and it is a receptor oncogene. And we'll talk a bit about this BCR ABL fused oncogene and how Gleevec can be used in treatment of such mutations. So the things we know from all this is that the characteristics of tumor cells are very distinct. There are different types of cancers, and we are convinced that cancer is a multi-step process, and there are tumor viruses involved in cancer, causing cancer. Oncogenes and tumor suppressor genes regulate the event or process of transformation, and proto-oncogenes can go bad because of mutations. And there are several cancer therapies available based on our understanding of molecular biology of cancer. At this point, are there any doubts?
Ma'am, at the end of the session, they will ask, ma'am.
Okay, that is fine. I will continue. We will go through some of the etiologic agents and their general mechanisms. Here you can see a normal cell can become a malignant neoplasm. All this we discussed right now because of chemicals, radiation, or viruses. So these are all DNA damaging agents which can act upon a normal cellular DNA and induce damage. If there is successful repair of the DNA, normal cell will continue its activity. Otherwise, we all know how P53 can protect the cell, or if it is damaged, it can induce apoptosis. More and more mutations will accumulate, and there will be several alterations in the regulatory genes, and there is inactivation of the tumor suppressor gene, activation of the growth promoting oncogenes, which will express altered gene products, and loss of several regulatory gene products, starting from growth factors, the receptors, signal transducing kinases, the transcription factors, and apoptosis agents, are all involved. Once the oncogene is expressed, tumor suppressor genes are inactivated, and other genes regulating apoptosis are altered. It leads to malignant neoplasm, and that is the clonal expansion leading to heterogeneity of the cell DNA. Now, environmental versus hereditary cancer, how environment plays a major role in causing cancer. Way back, people identified scrotal cancer because of the chimney sweeps. They used to get scrotal cancer. This is related to the occupation. So there is a relationship between occupation exposure to chimney soot and scrotal carcinoma. It was very well established. And these are chemical carcinogens present in the soot, direct acting carcinogens, which can activate or cause harm to the DNA. And carcinogens can also be indirectly acting. That means once they enter the body or the cell, they'll be metabolized, and active form of these carcinogens will then act on the cells. Most of them are electrophiles, like indirect acting carcinogens are activated into electrophilic species. So there are three main types of carcinogens: chemical carcinogens, physical carcinogens, and biological carcinogens. Some examples are given here. You are aware of many of them, like aflatoxins. You have heard of them: benzene, arsenic, aristolochic acid, etc., etc. This is a small list. The list is too big. I only chose some names which you may be familiar with. All right. Environment induced smoke or cancer is because of several chemical agents which are electrophilic in nature. Electron seeking is electrophilic, and molecules which bind to nucleophilic, that is electron rich molecules. So electrophilic carcinogens, they bind to macromolecules which are electron rich. They bind to DNA, RNA, and protein, causing damage, either mutations or altered proteins, to bring rise to or to give rise to cancer. Direct acting carcinogens, some examples, one or two, which you can remember, is nitrosomethylurea and benzyl chloride. They can directly act, need not be converted into electrophilic agents. Some carcinogens, which examples of indirectly acting carcinogens are PAH, that is polycyclic aromatic hydrocarbons, right? And chimney soot, we already gave examples: cigarette smoke, grilled meat, auto exhaust, that is vehicle exhaust, and so on. Some of the drugs commonly used are adriamycin, androgenic steroid, chlorambucil, cisplatin, cyclophosphamide, cyclosporine, diethylstilbestrol, right? Melphalan, tamoxifen, right? These are all commonly used drugs for treating cancers. Physical carcinogens are UV light, ionizing radiations like in X-rays, and asbestos. The spectrum you are very well aware of. Right? Skin cancer is one of the most common human cancers and one of the most preventable cancers. Right? Because of exposure to ultraviolet radiation. And asbestos is widely used in construction, insulation, and manufacturing. And it has a lot of silicates, right, which lead to cancer. This is one example of the asbestos. It can cause malignant mesothelioma. It is the cancer of the pleural cavity or peritoneal cavity. Latent period is decades. Ionizing radiation, high incidence of leukemia among radiologists. It was recognized way back in 1940s. And osteosarcoma, incidence in radium dial painters. So occupation dependent cancers, right? Now we go to the viral carcinogenesis. Viral infections account for an estimated 1 in 7 human cancers worldwide. Majority of these are due to infection with 2 DNA viruses. So these have the genetic material. DNA as the genetic material. You are very familiar with the human papilloma virus, right? And hepatocellular carcinoma causing virus, hepatitis B virus. Hepatitis B virus and human papilloma virus are the most common viruses which are known to cause cancer. Other viruses which are called oncogenic viruses, cancer-causing viruses, are Epstein-Barr virus, right? Herpes virus, hepatitis C virus, and virus which can cause AIDS. HPV, it is DNA virus with small double-stranded circular genome. Varying degrees of high and low risk viruses are there, depending upon progression of malignancy. Very bad slide to see, I'll skip. Epstein-Barr virus, African Burkitt lymphoma is caused by that, and B-cell lymphomas of immunosuppressed patients is caused by HBV, and some cases of Hodgkin lymphoma and nasopharyngeal cancers are caused by EBV, Epstein-Barr virus. These are small viruses, and they integrate their viral genome into the host genome. So integration is very important part here. As a consequence, viral proteins will be encoded and they will block the tumor suppressor genes. Helicobacter pylori is not new to us. It causes gastric infection and it is linked to gastric lymphomas and adenocarcinoma. And detection of H. pylori in majority of cases of gastric lymphomas is there. And there is antibiotic treatment to regress this type of tumor. There are several cellular mechanisms of carcinogenesis. Say in case of malignant lymphomas and sarcomas, there is a single gross genetic abnormality called as the translocation. Gross abnormality found in the chromosome. And that will activate the cellular mechanisms leading to malignant lymphomas and sarcomas. Otherwise, particular genetic alteration is there because of lots of heterozygosity or because of mutations, which is commonly seen in several malignancies and in several carcinomas. Genetic lesions in tumor could be activating or inactivating. These lesions could be dominant, recessive, or it could be due to somatic mutations or germline mutations. And the targets for mutations will be, as we discussed, oncogenes, tumor suppressor genes, and mismatch repair genes. Repair genes are very important. Damaged DNA has to be repaired. If the repaired genes are only mutated, then comes the great problem of cancer. So various mutations leading to cancer are gene deletions, amplifications, insertional mutation, point mutation, genetic instability because of microsatellite instability. These mutations sometimes are inactivating mutations, or it could be promoting mutations like epigenetic changes. Promoter hypermethylation will be there. Some mutations can lead to overexpression also of the genes. So they will, we already studied about oncogenes and tumor suppressor genes and telomere shortening effect leading to chromosomal instability, microsatellite instability. All instabilities because of genetic material will lead to cancer, and also the mutations in mismatch repair genes will lead to cancer. I won't go much about cell senescence and telomeres. Of course, they are very much related to cancer. You can see here, because of chromosomal abnormalities or anomalies, the chromosome can bridge, or it can fuse, it can translocate, or it can have breaks, single and double strand breaks, right, leading to chromosomal anomalies and ultimately leading to cancer. So the repair process, tumor suppressor genes and oncogenes are the major contributors for molecular biology of cancer. and they control the differentiation, control the role of apoptosis in our healthy and infected cells or derailed cells, and proliferation. Cell proliferation, apoptosis, differentiation are the major biological cellular events which are highly regulated and controlled. And if these are derailed, it will lead to the disease of cancer. Again, a pictorial representation of genetic instability contributing to cancer by activating mutations, overexpression of gene, amplification of the gene, deletions, inactivating mutations, and hypermethylation of the promoter cell. So every time this happens, it is a progressive acquisition. Step by step, neoplastic transformation will occur inside the cell with more and more mutations accumulating. Ultimately, what happens is apoptosis will be downregulated, cells cannot die, cells will go on multiplying, and there will be reactivation of telomerase enzyme. And self-maintained replication will be there in cancer cells. Longer survival rate will be there in cancer cells. We discussed in length about genetic instability in cancer cells. And cancer cells will now be able to induce a process called angiogenesis, which is formation of new blood vessels. And because of formation of new blood vessels, the cancer cells can escape from the different parts of the body by process of epithelial to mesenchymal transition, and they now will be able to invade and metastasize. So all the hallmarks of cancer are attributed to the major genetic instability which occurs in a cell to control the transformation process of transformation. So basic biological features of neoplasms are abnormal proliferation, angiogenesis, and invasion. These are the basics of the neoplasm. These are the features of neoplasm. However, differentiation, undergoing apoptosis, and senescence, these are the normal features which are highly controlled by oncogenes. So carcinogenesis is a multi-step process. Several genes have to be inactivated. Self-sufficiency in growth factors should be acquired. And self-sufficiency can be acquired by the tumor cells by activating the RAS oncogene. There will be insensitivity to anti-growth signal. Anti-growth signal means chemotherapy or agents which can inhibit cell division. These are anti-growth signals, and cancer cells will become insensitive to anti-growth signals, and that exemplified by loss of retinoblastoma tumor suppressor gene. And the cancer cells can evade apoptosis. They produce a lot of growth factor like insulin, like growth factor, IGF. This is a survival factor, and the survival factor will be more. As a consequence, death factors will be evaded, like evading apoptosis. Then there is limitation to replication potential because telomerase is activated. There will be sustained angiogenesis in cancer because of overproduction of a protein called vascular endothelial growth factor. Further, there will be tissue invasion and metastasis because of inactivation of cadherin protein, which is a cell adhesion molecule. Cell adhesion molecules will be altered. As a consequence, the tumor cells will separate from one another and they will start invading to other regions of the body, a process called as metastasis. So we have two different pathways which highly regulate our cells. One is a caretaker pathway, the other one is a gatekeeper pathway. And both the pathways are involved when there is deregulation and mutation leading to neoplasm. Say, for example, mutation of a caretaker gene allele will occur, then mutation of a second caretaker gene will occur, and there will be genetic instability. Say, for example, here I have given example of APC gene, right? Both the alleles will be mutated. As a consequence, there will be, because of additional mutation of the tumor suppressor genes, neoplasm will occur. This is another example where X-rays, oxygen radical, alkylating agent, and spontaneous reactions can lead to single strand breaks, and this excision repair mechanism will be hampered. UV light, polycyclic aromatic hydrocarbons, they can also induce, and as a consequence, there will be cellular transformation because of alteration in nucleotide excision repair. And here you can see the X-rays, anti-tumor agents, they all can induce stranded breaks in the DNA. Both the strands are broken here, and that can also lead to alterations in recombination, and recombination repair mechanism will be impaired and therefore leads to cancer. And replication errors will be there. As a consequence, you have mismatched. Here you see, instead of AT, AG will be there. Instead of GC, TC will be mismatch will be there. Insertion and deletion mutations will occur when there is replication errors leading to hampering of mismatch repair. So mismatch repair genes, recombinational repair mechanism, and nucleotide exchange mechanism, base excision repair mechanism, everything will be curtailed. As a consequence, a normal cell will be a tumorous cell. So deletion, amplification, mutation, insertional mutation, point mutation lead to genetic instability like microsatellite instability and chromosomal instability, contributing to cancer. The same is depicted, first mutation, second, and the third mutation. Mutations will go on accumulating from a single cell to several cells which have been produced because of higher cell divisions, leading to n number of mutations in a genetically unstable cancer cell. There will be bypassing of apoptosis. HTERD gene will be overexpressed and it will be very active. BCL2 overexpressed. You see, the tumor suppressor gene is knocked off. And cell adhesion molecules like cadherin, it will lose its ability to adhere one cell to the other, and cell cycle deregulation will be there because of activation of oncogenes and inactivation of tumor suppressor genes. So net growth is there if proliferation rate exceeds the rate of death. So when growth is more, death will be less. So death rate will be very low when growth is more. And growth is more when there are more growth factors, when there are more growth factor receptors expressed, when there is mutation on RAS oncogene, and when there is activation of the retinoblastoma gene, there is increased proliferation contributing to the process of cancer. Here you can see a normal epithelium becoming a metastatic type of tumor when there are so many changes in the epithelial cells. The APC gene will be altered, leading to dysplastic aberration crypts, because we are talking of colon cancer now. There will be early adenoma, intermediate adenoma, late adenoma. All these will show changes in the tissue, in the colon tissue. So if there is a biopsy taken by colonoscopy, the doctors and pathologists can identify changes which occur in the tissue. And later on, if the biopsy is, from that DNA will be isolated, and it can be sent for next generation sequencing. And there, scientists will identify that there are mutations on the RAS gene, mutations on the RAF gene, and the SMAD pathway, signaling pathway, is derailed because of factor mutation and mutation in P53 and mutation in the protein related to liver. So many mutations can be identified during the process of colon carcinoma, leading to metastasis. Therefore, the current view is that multi-stage cancer is a multi-stage process. It involves inherited and somatic mutation of the genes. I covered all this, so I will skip that portion. A proto-oncogene is converted to an oncogene. There are several proto-oncogenes which are identical. It is regulated because both the alleles are normal. The oncogene is not mutated. But here, even if on one allele it is mutated, there is no regulation on the speed of cell division. Likewise, these oncogenes, they activate many types of cancers. You have to remember the names of these very, very important oncogenes: RAS oncogene, MYC oncogene, RET oncogene, MET oncogene, the CDK oncogene, BCR, ABL2, and BCL2. At least half a dozen of them. Second requirement is that second mutation should occur to cause this particular effect. And here are some examples of tumor suppressor genes like retinoblastoma gene, WT, Wilms tumor gene, neurofibromatosis type 1 gene, NF1, NF2, and DCC gene is involved in colorectal cancer. BRCA1 and 2, BRCA1 and 2 are involved in breast cancer. These are tumor suppressor genes. And if they are mutated, you cannot suppress the growth of the tumor. Already I gave you a glimpse of how the retinoblastoma gene will act, and further we will see about P53 and APC gene, which is involved in other types of cancers, Tumor suppressor genes, the key attributes are they are involved in familial cancer syndromes and inactivation in common human cancers. These common in human cancers, many types of cancers, they are inactivated by process called as loss of heterozygosity. They are recessive at cellular level. And I told you the two alleles have to be knocked off. That is called the two-hit hypothesis. In order to normal tumor suppressor gene to be knocked down, two-hit hypothesis is required. Retinoblastoma and two-hit hypothesis. It is a gene responsible for tumor in retinal stem cells. And average age, you can see it is in months, means newborn babies, infants are more prone for retinoblastoma. 1 in 20,000 live born infant is afflicted with retinoblastoma. Male and female kids, both are equally affected. It is likely to be bilateral, means familial. From parents, they can inherit. This particular hypothesis is called, based upon finding by a scientist called Knudson's two-hit hypothesis, where first hit is a mutation. It is present in the germline. And because it is in the germline, it will be in all cells of the body. But this first hit of mutation is not sufficient to promote a normal cell to become a tumor cell. Though it is in all cells of the body, if a person is carrying it in the germ, because the mutation is in the germline, the second hit to inactivate the remaining allele is necessary to promote tumor formation. This gives, the presence of a mutant already in the body, in cells of the body, is already a risk. So the likelihood of somatic mutation will occur at least one retinal cell during the development. The dominant inheritance pattern of transmission could be explained. So illustrates how inherited and somatic genetic changes might collaborate to give tumorigenesis. Because the first hit was already got from germline, from parents, they already carried, one of them already carried the mutation in retinoblastoma gene, and during the life of that person who is carrying already mutated gene in a somatic cell, A second chance of mutation is very likely, leading to tumorigenesis. So this is a cartoon representation, I, where two retinoblastoma genes are intact copies. But here you can see there is a predisposition because of the germline mutation. Only one intact copy is there, the other one is mutated. Now you can see during the development, that is the infant phase, second copy may also be affected. Loss of both copies will give rise to the cancer of the eye, that is retinoblastoma. And already we discussed how retinoblastoma acts. It is a mutation in the E1A protein. E1A has the ability to bind to retinoblastoma, also inactivated E1A's transforming function. I won't go into the details of it. And retinoblastoma protein is regulated by phosphorylation. We discussed this, that it binds to the elongation factor. When it is not phosphorylated, it binds to the elongation factor and it will repress gene expression. When it is phosphorylated, its binding to the elongation factor will be removed. As a consequence, there will be transcription and translation, giving rise to genes which are involved in S phase of cell cycle division. P53 is a best example, again, like retinoblastoma gene of a tumor suppressor. And to maintain a normal phenotype, it is very, very important that this gene is not mutated. And again, a viral study led to the identification of P53 gene. Where is it located? It is located on chromosome number 17 on the P arm of the chromosome. And it has 393 amino acid. Why I want to tell you this is it has different domains in the structure of the protein. Transcription activation domain, because it is a transcription factor. DNA binding domain, it has to bind to DNA to induce transcription, and it has oligomerization domain. So it can oligomerize. So this DNA binding domain is the one which is vulnerable for mutation. Cellular stress, that is DNA damaged by UV rays, ionizing radiation, or chemical agents, or oxidative stress, will regulate the P53. These will activate protein kinase. Protein kinases can phosphorylate the N-terminal domain of P53. When it is activated, P53 will be released from this MDM protein. Otherwise, it is bound to MDM protein and it is inactive in this state, in the bound form. Phosphorylation of P53 will activate P53, and P53 half-life increases and conformational change will occur. Otherwise, P53 alone is very unstable protein. It will be degraded by proteasome. But upon phosphorylation, the conformational change will induce stability to the protein. The MAP kinase family of proteins are the ones which are involved in phosphorylating P53, because the MAP kinase family responds to stress. And there are other kinases, the checkpoint kinases, which are involved and they respond to DNA damage. Apart from the MAP kinases, these kinases can induce the phosphorylation of P53, so disrupt the binding between P53 and MDM2. And as a consequence, there will be conformational change due to phosphorylation on P53. Now it can act as a transcriptional co-activator in a very huge multi-complex protein transcription factor called P300. and thereby it can activate or repress expression of several genes. However, mutations on p53 will occur and mutations will deactivate p53, especially in cancer, and this mutation will occur in the DNA binding domain of p53. Most of the mutations will destroy ability of protein to bind to the DNA sequence, and it will prevent the transcription activation potential of p53. And these mutations are recessive loss of function mutations. And the molecules of p53 with mutation in the oligomerization domain dimerize with wild type p53. So wild type and the mutated p53 will oligomerize and prevent from activating transcription. Therefore, oligomerization domain mutations have dominant negative effect on the function of p53. There is loss of function mutation on the DNA binding domain, and there is a dominant negative type of effect on the function of p53 when oligomerization domains are mutated. So the basic function of transcriptional activity of p53 will be impaired on mutation. p53 has many anti-cancer properties. When it is not mutated, it can activate DNA repair genes, repair proteins, DNA repair proteins will be activated. It can also hold the cell cycle at G1 to S regulation, because we discussed about the P21 inhibitor of cyclase, that is cyclin dependent kinase. It can initiate apoptosis. It can repair the DNA. It can arrest the cell cycle and regulate the cell cycle, and it can initiate apoptosis. These are all very favorable function for anti-cancer property of p53. However, when it is mutated on either the DNA binding domain or on the oligomerization domain, it can be inactivated, and this p53 tumor suppressor gene will be no more functional, leading to carcinogenesis. I have already explained the function of p53. Next important tumor suppressor gene besides retinoblastoma and the p53 is the APC gene. APC stands for adenomatous polyposis coli. It is activated in adenomatous polyposis, and there will be 100 to 1,000 of benign tumors called as polyps in the colon. Small round polyps will be there in the colon, but however, phenotype is that they are benign and evolve invariably, they become invariably cancerous with due time because of the process called Knudsen two-hit hypothesis. Familial adenomatous polyposis, already in germline DNA, one mutation will be there, and the second hit APC is also mutated. Because of second hit, there will be second mutation, and therefore it will lead to colorectal cancer. And major player in this is the Wnt signaling pathway. We discussed about the MAP kinase pathway, growth factor related pathway, RAF, RAS, and all that. In this APC, it is the Wnt signaling pathway which is playing a major role. I won't go into the structural aspects of APC or the Wnt signaling pathway. APC gene, when it is mutated, more than 800 mutations are seen in APC gene. So this number you can see is very enormous. So these are the phenotypes which can be seen in the colon. So inside this colon, which is, you can see the intestine, polyps will be there in the normal colon, and these polyps will become cancerous when there is a two-hit hypothesis, when the second allele is mutated. So you can see in colon tumor progression, a normal colonic epithelial cell. Because of inactivation of both copies of the APC gene will get to become early adenoma, and there will be a dominant mutation in the KRAS gene. RAS is a very, very important gene which is involved in signal transduction. It encodes a protein kinase, and if that is mutated, you get intermediate adenoma, followed by late adenoma, where both copies of DCC gene will be mutated, and still benign stage of colon cancer. Lot of hundreds of polyps will be there, but this polyp will turn to be malignant adenocarcinoma because of loss of P53 gene. Now P53 gene will be mutated and it will be lost. Further mutations on other genes will cause the malignant adenocarcinoma to become metastatic adenocarcinoma, and metastatic tumor, ultimate, it is fatal. Like other tumor suppressor genes, the DNA repair genes are very, very important. If they are mutated or inactivated, they are implicated in broad spectrum of human cancer, because genetics or the mutations are the major reasons as a causative of cancer. The inactivated mutated gene repair becomes very, very important pathway in order to regulate normal growth in a cell. Inactivation of such repair gene may increase the mutation rate in other cellular genes, including proto-oncogenes or other tumor suppressor genes, leading to cancer. As a consequence, the progression of tumor will be accelerated. More the mutations on repair gene, fast is the growth of the tumor. Some of the repair gene examples of nucleotide excision repair genes, mismatch repair genes, and genes involved in somatic mutational disorders. I did explain to you about nucleotide excision repair. Adducts will be formed by carcinogens like benzpyrene and UV light. You have heard of adducts which are formed, the thymidine dimers and so on. And these individuals, they are vulnerable to UV light. And if repair is not there, it results in skin cancer. Likewise, in mismatch repair, if the repair is not there, it will lead to colon cancer. Just now I showed you the example of how several gene mutations leads to colon cancer if mismatch repair is not there. So a lot of mismatch will be there, and it will lead to mutations like normal DNA repair will not be there, mutations will be impaired. In concluding about this, tumor suppressor genes control key steps in various cellular processes. Control of cell division will be there by tumor suppressor genes, example, retinoblastoma and P53. P53 responds to stress, and APC control signaling pathway. DNA damage repair genes, they have a major role in controlling the transformation of a normal cell to a tumor cell. So identification of tumor suppressor genes and the oncogenes and the pathways involved in the mechanism of action of these genes, they have provided targets for the development of new therapeutic compounds for cancer. So is that okay for today? Can I stop now?
Yes, ma'am. Yes, ma'am. So now the session is open for a discussion. Ma'am?
Yeah, sure.
Ma'am, already some of the participants raised some questions. I'll read one by one. Can I read, ma'am? Shall I read now?
Sure.
Ma'am, first question is,
Yeah.
What is the difference between physiological and pathological cell death?
Physiological cell death is the normal renewal of the skin. Example, cells in the skin will grow and will die, and these are all controlled by apoptosis. Right? Normal physiological death is in normal cells and it occurs normally. Pathological death is because of infection, because of inflammation, because of cancer. You can induce the death in cancer cells, right? And that is pathological death of cells. Because it could be due to viral infection, bacterial infection, cells can die, inflammation, cells can die, right? Reactive oxygen species can induce cell death, right? These are all very highly regulated processes, right? Like necrosis is again a death, but necrosis is not controlled like apoptosis. So programmed cell death is there in our body. Self-renewal is there in our body.
Okay, madam. And the second one is, what are the major symptoms of liver cancer and which method is effective to detect it?
Symptom of liver cancer, liver has a very important role to play in our body. The major function of liver, how hepatocytes and hepatic functions play a role in metabolism of macromolecules in our body. So diet and liver are highly connected. And liver cancer can be identified by malfunctioning of the liver, indigestion problems, pain in the liver. You can feel the pain in the body. Liver will degenerate, right? It cannot regenerate once it has cancer.
Okay, ma'am. The last one is whether normal cyst during surgery have changed to turn as a cancer.
Usually normal cyst during surgery won't turn into a cancer, right? Normal cyst can be removed. Because of surgery, it cannot turn into cancer. So cyst can be easily removed. Suppose there is a cyst in the breast, it can be easily removed. It should not be left there. It may overgrow, right? It is better to remove. And removal does not cause cancer. Removal will stop the growth of the cyst there only, right? Only with time, if that cyst becomes bigger and bigger, if it is infected, if it is inflamed, and if there is again mutation, more and more mutations in the cells of the cyst will turn a normal cyst into a cancerous cyst. And also in case of liver cancer, identification, usually, several good methods are there for identification. I forgot to tell you that, right? It can be identified by scanning, CT scanning and so on. MRI, CT scans are very useful.
Okay. Now the next one is, all tumor are cancerous cell.
See, these synonyms are used, tumor and cancer. Several tumors or cancers are not malignant. They are benign tumors. Benign tumors or benign cancers are not dangerous. They will be present in one place. They will keep on growing bigger and bigger and bigger. They won't affect other parts of the body. Wherever they are, they grow. But once a person sees there is an extra growth, they will go and get it tested, and pathologist will find out by biopsy. Doctors will tell you that it is a benign tumor. It is not a malignant tumor. It is not spreading type of tumor, because doctors will identify that there are no tumor cells in node. Nearby node will be tested. In my talk, I told you, if it is node negative, it is a good prognosis. Means patient is not in trouble. All right. The prognosis will be good if it is node negative. If there is a benign tumor and nearby node is not having any tumor cell, that means it is not a spreading type of tumor. Only when the tumor is malignant and a spreading type through blood vessel and through lymphatic vessels, it will spread to other parts of the body. Then only it is dangerous.
Okay, the last one is, ma'am, the house microsatellite instability cause cancer?
I just now explained a lot of microsatellite instability because of mutations in the genes, and also the telomerase activity is very, very important, right? Microsatellite instability is basically because of mutations. It can be different types of mutations like point mutation, deletions, and so on. Any more questions,
Okay,
Dr.
ma'am.
Durai?
Ma'am, I think the questions are listed.
Okay.
Ma'am, once we remove the tumor in breast, whether there is any chance to produce tumor in the same place?
Yeah, there are, right? If breast is removed,
Yes, madam.
if breast is removed, usually breast will be removed if there is a bigger tumor. Means if the tumor, there are chances that other breast can also get cancer, right? That is why they remove. And if it is a very small tumor, whole breast will not be removed, right? Only that portion will be removed. Whole breast will be removed. In breast also, there are different types of tumors, ductal carcinoma and so on. So if the tumor has spread to different parts of the same breast, whole breast will be removed. And besides, the node which is under the arm, all nodes here will be removed under the arm. And chances are there the second breast may also be removed, though it has still not got any cancerous cell, to be on safer side. Person can still be alive for long duration, 15 years or more, if caution is taken by surgery. And nowadays, there is reconstruction just to give the aesthetic look and so on. So one should not get worried. Life is much more good and long. Even if two breasts are removed, one should not get worried about it.
Okay, ma'am. Thank you. I
In
thank the
tomorrow's lecture,
participants
you will,
to make
yeah.
Make the session is interactive. Ma'am, yes, ma'am. You want to share anything, ma'am?
Yeah, in tomorrow's lecture, you will hear more about the therapeutic aspects. In tomorrow's lecture, you will hear more about therapeutic aspects.
Okay, ma'am. Okay. So now in the end, thank for your wonderful present and introduction and sharing knowledge about cancer. Thank you, ma'am. I thank the participants to keep patient and listening and made the session is highly interactive. Thank you, participants.
Yeah, thank you.
Now the session is end. We wind up. Tomorrow we'll meet by 9 o'clock. Thank you, ma'am.
Thank you.
The link will be shared with the email and group before the session start. Thank you.
Thank you, ma'am. Meet you tomorrow through virtually, ma'am.
Yes, thank you. Bye.
Okay, ma'am. Thank you, ma'am.
Thank you. Thank you, ma'am. Thank you so much.